会议呢，基本上是一个科学的会议，是一个有关科学发生的历史。
副教授是 JAK-STAT 信号通路的重要发现者之一，他的科学成就在国际上获得了广泛的认可。
今天的会议隆重庆祝 JAK-STAT 发现30多年来取得科学进展和成就。
我很高兴啊，接受傅老师的邀请，解决未被满足的临床需求，应该说呀，发挥了重要的作用。
Signal transduction and activators of transcription.
Yeah.
STA.
I hope I can see the slides. I'm going to have to ask you to change them for me, please.
We published the first analysis.
So I'll do my best to give my talk remotely in California.
And mechanisms of signaling and physiological fine-tuning.
Virus receptor discovery and development of vaccine antibody.
而这个交往更多的是在胁迫感染这种情况下发生的一个死亡。
我只是一个协调的人，所以感谢我们的团队。
傅老师说这个 JAK-STAT 的信主，我说我也做过这个 JAK 吧。
主要给大家也是分享一下，就是说这个今天是借此机会嘛，就庆祝那个 JAK-STAT 通道发现的30周年。其实理论上讲还是
抑制性特别强的一个肿瘤，它因为抑制性很强呢，
感染它应该是按照不同的这种它的病理生理改变去理解。
是 Lancet 刚刚在全球做了一个统计。
First thing I sent to Jim Darnell, saying, Jim, look, I got this SA2 domain. That's great discovery. Can explain our mechanism now.
Okay, thank you. I hope I can see the slides. I'm going to have to ask you to change them for me, please. So you have control of the slides, I think.
Yes.
Great.
Yes.
Okay. So it's a great pleasure to be able to talk at this wonderful meeting, and I'm going to be talking, who asked me to talk about some of the history, and I'm also going to be talking about some of the major things that have happened in my lab over the last 20 or so years, and finish with some new data that isn't published. It's focusing on unexpected and novel roles of U-ISGF3 and U-STAT2. The U means unphosphorylated, means these factors have no tyrosine phosphorylation, and yet they function very importantly. Can I see the next slide, please? Oh, we skipped one. Yeah, thanks to Fu and the other organizers for inviting me to speak. And I feel so badly that as things turned out, I couldn't be there in person. I had intended to come, and for a variety of reasons, it didn't turn out to be possible. Okay, next slide. So when we started to look at what was responsible for interferon-dependent signaling, we decided to try to use a mutational genetic approach to approach the problem. And the question we asked was, could we get mutant cell lines that lack genes encoding interferon signaling proteins? Now this is the late 1980s, so nobody had dreamed about CRISPR technology. So there was no really good way to get cell lines lacking expression of any protein. But we decided to try, and we set up cells that could be selected with an interferon responsive promoter which encoded a lethal selectable marker. To get efficient mutation, we used a frameshift mutagen called ICI191. We wanted to each chemical event to hopefully lead to a loss of function. And we did a pilot experiment just to look at the feasibility. So we mutated the single copy HPRT gene, for which there was a good selection, and found that we could, under specific conditions, it could be eliminated at a frequency of about 10 to the minus 5. So we would then predict that 2 copies of a gene would be eliminated at a frequency of about 10 to the minus 10th, the square of 10 to the minus 5. I can't see the whole slide. Can you get me down to the bottom of the slide a little better? No, not the next slide. I can't see the bottom of this slide. Just go back, please. Can we go back? Yeah, okay. Okay, next slide. Okay, so if we guessed that there were 10 different proteins that are required for interferon signaling, we would need to screen about approximately 1 billion cells to get 1 mutant cell line, and that is what we did. Fortunately, it worked with a lot of hard work. Can I see the next slide? So in the late 1800s, I'm sorry, late 1900s, 1989, we published this paper together with Ian Kerr. Sandra Pellegrini was the lead author, showing that this could work. We could obtain mutants by random mutagenesis that were unresponsive to interferon. The next slide. So, Sandra Pellegrini left my lab at the Imperial Cancer Research Fund and established her own lab in Paris at the Pasteur Institute, and she did the very hard work of complementing one of our mutants with genomic DNA, it turns out, with a cosmid. And that led to the identification of TYK2, a previously known kinase, but that had no known function. And this paper was really crucial in teaching us that the kinase TYK2 was essential for type 1 interferon signaling. Next slide, please. So we needed more mutants, and we found that just simply by increasing mutagenesis, by increasing cell killing, we could get a lot more mutant cell lines. We could use hybridization to sort them into complementation groups. We obtained, in the end, 8 different complementation groups. And with the help of clones from the Darnell lab and others, we found we were able to complement all of these mutants. So we have 8 mutants which lack the expression separately of JAK1, JAK2, TYK2, STAT1, STAT2, IRF3, I'm sorry, this should be IRF9, and one subunit each of the IFNAR and gamma receptors. We then published 8 papers with Jim Darnell's lab in which we used their constructs and mutations of the constructs to learn about the signaling pathways. These were the only cells available for quite a few years that lacked expression of these proteins, and we sent these cells to well over 500 laboratories, I might say without charge, which helped to define many different JAK-STAT pathways. And I think many of those cells are still in use. Well, I probably don't need to remind you of this pathway, but just to be complete, here's the type 1 interferon signaling pathway, the 2-component receptor, TYK2 and JAK1. ISGF3 is a trimer composed of phosphorylated STAT1, tyrosine phosphorylated STAT1 and STAT2, and IRF9, and that drives the expression of ISGs by binding to the response element in interferon-stimulated genes. Next slide. So here was a surprise. When we looked, this is a Western blot, when we looked at a long time course of expression of STAT1, STAT2, and IRF9, the 3 proteins that comprise ISGF3, we saw there was a major increase in the protein expression in 2 different human cell lines of STAT1, STAT2, and IRF9. And this was very dramatic, and it told us that these 3 genes were themselves ISGs and that they responded to interferon. But we wondered, what in the world was this expression of what turned out to be proteins lacking tyrosine phosphorylation in the case of STAT1 and STAT2? What was that for? Next slide. Well, we found that simply expressing these proteins, even in the absence of interferon treatment, just from constructs, could give cells that were resistant to DNA damage and resistant to virus infection. So they retained about 3 logs of resistance to virus infection compared with many logs for cells that contain the immediately phosphorylated proteins. Next slide, please. And this correlated with wonderful work done by Ralph Weitzelbaum, who looked in many different tumors and identified a pattern of interferon dependent gene expression that he called the IRDS, interferon response. And in the next slide, so I'm sorry, this correlated with resistance to DNA damage and ionizing radiation. So we looked in cell culture now at the ability of how cells responded to long-term treatment with a very low amount of interferon. So this is half a unit of interferon applied every other day. And you can see that there is, even in response to this little bit, phosphorylation of STAT1 in a short time. But if we continue this over quite a couple of weeks, we lose the phosphorylation of STAT1, but we see the big increases in steady state now expression of the 3 ISGF3 components. And so this explains how in cancer, constant exposure to a low amount of interferon in the tumor can induce this pattern of expression, which comprises only about 20% or so of the genes that are regulated in the primary response. And this, what we saw in cell culture, correlated almost perfectly with the pattern of expression that Weitzelbaum saw in tumors, in patients. Next slide. So this is kind of a summary of where we think things are. In response to a high dose of interferon, ISGF3 is formed with the phosphorylated STATs and drives the expression of all of the interferon responsive genes. And these can be sorted into 2 categories. Those that have an ISRE element and respond only to the phosphorylated form, and those that have a related element, which we call the UISRE, which responds to both the phosphorylated and the unphosphorylated form. So over time, when the negative regulation, especially the induction of SOX1, shuts off the phosphorylation, we're left with UISGF3, no tyrosine phosphorylation, which still constitutively drives the expression of this subset of interferon-regulated genes. And according to Weitzelbaum, these have important roles in maintaining maintaining resistance to DNA damage and ionizing radiation in cancer. Next slide. So here's another very dramatic result. We wondered whether expression of self-interferon was important for the survival of cancer cells. So here's an example of a cancer cell line in which we simply knocked out the interferon gene, leaving everything else intact, the interferon beta gene. And those cells do not survive in tissue culture. Here's cell death compared with control cells. But if we supplement the medium with exogenous interferon, we can largely rescue that death phenotype. So we conclude that some cancer cells, there is another example that will be published, some cancer cells absolutely rely on their ability to produce their own interferon in order to survive. They are addicted to interferon, type 1 interferon. Next slide, please. And part of the mechanism by which this works is by an effect of PD-L1. Well, you all know about PD-L1 for its role in binding to and inhibiting the activity of CD8 T cells through its interaction with PD-1. But this is a cell intrinsic function of PD-L1. PD-L1 is a membrane protein, and in the complete absence of any other cell line, it is effective in sustaining the ability of cancer cells to respond appropriately for them to type 1 interferon. Next slide, please. So here's an experiment in which we're treating cells with a lot of interferon, and these cells either have, this is a time course, these cells either have PD-L1 or it's been knocked out. And you can see when PD-L1 is there, there's detectable but low-level activation of STAT1 phosphorylation. But when PD-L1 is lost, there's greatly enhanced activation of STAT1 and STAT2 phosphorylation. And, so we conclude that somehow cell surface expression of a high level of PD-L1 partially blocks the action of interferon on cancer cells. And we call this the Goldilocks effect. Cancer cells like a little bit of interferon, which sustains their ability to express the subset of proteins that enable them to resist DNA damage, but they don't like a lot of interferon, which inhibits their growth. So they modulate the amount of interferon. And the paper that I just showed you goes into more detail about additional effects of PD-L1. We don't yet know the mechanism. We suspect that there's probably a direct interaction between PD-L1 and the interferon receptor, but we haven't shown that yet. That's just a hypothesis. Okay, next slide, please. So now I'll talk about U-STAT2, that's STAT2 lacking tyrosine phosphorylation, and novel functions in infection. Next slide. So U-STAT2 lacking tyrosine phosphorylation is nevertheless phosphorylated, but not on tyrosine. It's phosphorylated on 2 threonine residues, which we found by mass spectrometry. And those residues are threonine 387, which is phosphorylated by CDKs and other CDK9 and other CDKs, and that's a constitutive phosphorylation. And there's a phosphorylation on a nearby threonine 404, which is catalyzed by IKK epsilon and RIP2. These are kinases that are activated by viruses and bacteria. So this is an inducible phosphorylation. And it turns out that these 2 phosphorylations have important biological effects, but in opposite directions. Next slide, please. So they affect, I can't see the top of the slide, but the phosphorylation actually facilitates the formation of a very interesting complex between unphosphorylated STAT1 and STAT2, each of which lack tyrosine phosphorylation. And this phosphorylation on threonine 387 facilitates the formation of this complex, which is an inactive inhibitory complex. And the phosphorylation of 404, in contrast, opposes the formation of this complex. Next slide. So the phosphorylation of 387, which facilitates the formation of the inactive complex, inhibits type 1 interferon signaling, and this is a diagram just to show that. Next slide, please. And conversely, the virus-induced phosphorylation of STAT2 in these tyrosine unphosphorylated dimers boosts antiviral defenses. It activates interferon signaling. And that's 404, the 404 residue. Next slide, please. So, Xin-Yuan, who's responsible for a lot of the work, Xin-Yuan working in Jinbo Yang's lab in Qingdao, generated STAT2 knock-in mice in which the 2 threonines were substituted. There's a slight change in nomenclature. The numbering is slightly different in mice and humans. There's one residue different. But anyway, this 403 residue in mice when converted to alanine, which can't be phosphorylated, of course, impairs the interferon response, and the corresponding phosphomimetic mutation improves the interferon response, and in contrast, the phosphorylation of this threonine residue also, the impairment of the phosphorylation of 386 also improves the interferon response. Next slide, please. So, referring now back to a very nice paper published from Uwe Nickelmeyer's lab, showing that STAT2 is a pervasive cytokine regulator because it inhibits STAT1 function in multiple signaling pathways. So, as I mentioned already, in the absence of tyrosine phosphorylation, STAT1 and STAT2 form a very stable, in Nickelmeyer's hands, this is a 10 nanomolar stability dissociation constant, heterodimer. And consequently, when one overexpresses STAT2, the activation of STAT1 is impaired, and the interferon gamma response doesn't work because all of STAT1 is tied up in this inactive complex and none is available for activation in response to interferon gamma. Next, please. So we decided to try to understand the basis of all of this at a structural level. And in order to do that, Xin developed a very, very nice method to isolate cells specifically from tissue culture, to isolate from other complexes, specifically from tissue culture cells. So in this method, STAT1 is tagged with 1/2 of a fluorescent protein, venous protein, and STAT2 is tagged with the other half. When these two bind to each other, the complete venous protein is generated and generates a fluorescent signal in the cell. So cells can be sorted that contain this complex. But then the complex can be physically isolated by binding to a bead that contains an antibody which is high affinity for the complete venous protein. Following the binding and washing, this whole tag can be cleaved off with an engineered TEV protease site. So what one is left with, eluting from the column, is STAT1 and STAT2 complex, not the individual proteins, but only the complex, from the mammalian cells, carrying whatever post-translational modifications happen in the cell. And this complex was used in collaboration with Derek Taylor's lab at Case Western Reserve University to determine the complete high-resolution three-dimensional structure of this USTAT1, USTAT2 complex. And that led to really clear understanding of the effects of the two threonine phosphorylations. Next slide, please. So in the cryo-EM, we can see at atomic resolution the phosphate bound to threonine 387, and it sits next to a positively charged lysine residue in USTAT1, which is here. And that charge-charge interaction in a hydrophobic environment helps greatly to stabilize the inactive complex, 10 nanomolar complex between the two proteins lacking tyrosine phosphorylation. Next slide, please. Now, the formation of this complex, it turns out, has importance for understanding why dominant positive mutations of STAT1 cause disease. So, candidiasis is a disease that is caused by relatively rare mutations in STAT1, which cause hyperactivation of STAT1, and through mechanisms that Jean-Laurent Casanova's lab have worked out very well, leads to candidiasis through interference with IL-17 production. So these individual mutations, all of which are rare, cluster in many different patients to this region of STAT1. And we can look at where these mutations are in the 3D structure that we have. Next slide, please. So, one of the mutations in STAT1 is the loss of this positively charged lysine residue, which is substituted by an isoleucine. And that prevents the charge-charge interaction, which stabilizes the dimer and leads to destabilization of the dimer. When the inactive unphosphorylated dimer is destabilized, both STAT1 and STAT2, the levels of those increase, and they could be, this regulatory part of the control is lost, and you get too much STAT1 activation. Many other residues that sit in the interface between STAT1 and STAT2 in this complex have also been shown, when mutated, to lead to hyperactivation of STAT1 and candidiasis in these rare patients. So I think we now are beginning to get a molecular understanding of the basis of why these STAT1 gain-of-function mutations lead to disease. There are other mechanisms to be sure, but at least part of the story has to do with the dysregulation caused by lack of control through this inhibitory. It's a very important inhibitory complex. Next slide, please. So, USTAT2, involving threonine phosphorylation, has other important functions. So, one of the pathways where it does have an important function is shown in this recent publication with Xin-Yuan as a major author here, and it involves inhibition of trafficking of STING. So, as I'm sure you know, cytoplasmic DNA activate cyclic GA synthetase, which makes the cyclic dinucleotide CGAP, sorry, which then is an activator of STING. And when STING is activated, it normally moves from the ER to the next compartment shown here, where it signals to NF-kappa B. But in order to signal in order to signal interferon synthesis, it needs to move to the late endosome. And that movement is impaired when STING forms a complex with STAT2, with USTAT2, lacking tyrosine phosphorylation. That interaction depends, however, upon the phosphorylation of threonine 404. So in the 404 T to A mutant, this inhibition of just the interferon synthesis part of the STING downstream pathway no longer happens. And interferon is hyperactivated in response to cytoplasmic DNA. We've seen other regulatory roles of USTAT2, which I'm not going to go into in detail. We also published a paper showing that the USTAT2 IRF9 complex facilitates the activation of a subset of NF-kappa B dependent genes by binding to ISRDs that are present in the upstream region of the promoters of these genes. And there's more to the story than that. So the bottom line is that STAT2, lacking tyrosine phosphorylation, has a completely novel set of functions, quite separate from its signaling, well-known signaling functions in interferon, and is an important regulator of several different important signaling pathways. I think that's the last data or information slide. Can we have the next slide, please? So I just want to finish by thanking the people who contributed so importantly to this work, especially Xin, who now has his own lab at the Cleveland Clinic, Hanju, who did the work on the UISGF3 story, and who now has her own lab in the Karmanos Cancer Institute in Detroit, Xin-Yuan, who's now happily working in my lab, happily for me, I hope she's happy as well, together with Elise, who runs the lab. And many thanks to Derek and Wei, who are continuing to collaborate with us and continuing to look at 3D structures of complexes that we can isolate using this novel technology directly from mammalian cells. So again, thanks for your attention, and I'll be happy to try to answer any questions you may have.
Okay. 就是现在呢，我们要成立这样的一个研发联盟，这个也是为下次会议做准备的。